It's a great pleasure to introduce our speaker, Professor Dr. Sylvia Moraes, an expert in plant genetics and plant-soil microorganism interaction with a strong focus on plant response to biotic and abiotic stress. Dr. Sylvia Moraes holds a PhD in genetics and molecular biology from Unicamp and is current research at Embrapa and a faculty member in the graduate program in bioengineering at Federal University of São João del Rei. Since 2024, she was served as principal research at the Research Center for Genomics Applied to Climate Change, a collaboration between Embrapa, Unicamp, and FAPESP. Her work has made important contributions to plant physiology, molecular biology, microbial ecology, and nutrient use efficiency in grasses. We are honored to welcome her today and looking forward to her lecture. Today, Dr. Sylvia Moraes will present the lecture entitled From Genomes to Fields, Omics-Based Strategy for Efficient Nutrient and Water Use. Please welcome Dr. Sylvia. Dr. Sylvia, thank you for joining us today. You may now begin our presentation. Remind to everyone, we will have a questions and answer session after the lecture.
Thank you so much to be here. So I would like to start actually thanking the group. It's a really great pleasure to be here. I don't know if everybody knows, but Dr. Venkovsky was the head of Embrapa Maize and Sorghum for 5 years in the past, and his contribution and vision enriched a lot our unit at that time. So it's really a pleasure, and I'm really happy to see a nice group of students, organizing such a great meeting. So congratulations to the professor that is guiding, Dr. Beldin, and everybody. Okay. So I'm a researcher at Embrapa Maize and Sorghum, that is in Sete Lagoas, Minas Gerais, but I'm also now at Campinas. So we have a joint center there. There is a Genomic Applied to Climate Change Center. That is a joint unity between Unicamp and Embrapa. So, food is essential for us, right? I mean, everybody needs food at certain point. So we have been selecting food since like the beginning. So it's very easy when, people are seeing like a nice fruit and a more or less fruit, you're gonna go for the nice one. You can show this to a kid and it's gonna be there. So it's a natural process that happened. But when we saw now the work that came from Gregor Mendel and other genetic scientists that came after that, we start actually to analyze and select these in a more conscious way, because we were trying to understand, the genes and what was going there. And after the discovery of DNA, the genetic code, we have enhanced a lot our knowledge about it. So it's after like the sequencing and how we apply and breed for that. So when we have a scenario of climate change, growing population, you have a need for food. So we need to produce more and with quality. So that's essential for us in general. So what's been happening now, not now, but for the breeders, is that we have a trait that we desire, for example, tomato flavor, but we also want to have a gene resistant for a certain disease. So we need to breed, of course, this. But all the breeders know that you need a lot of cycles, in order to get to a more resistant and flavor fruit, for example. So what will happen? So if we use genetics, molecular biology tools, and other stuff like gene editing, transgenics, is that we can accelerate this process or insert a gene that was not that before. So that's a great way that we can accelerate and have different options. So with that, we can enhance what our ancestors were doing like thousands of years ago. We had at the mid of last century a big crisis, let's say, that we kind of were saved by the Green Revolution that was led by Norman Borlaug, but among different scientists, when we have different technologies being applied, and that was a great contribution for food production. So here in Brazil, we have the Cerrado region that if you think about 60s, 70s, there was almost no production there. So even Norman Bullock said that it was amazing how such a soil that was too acidic and too poor in nutrients could produce, food. So nobody was actually believing. So my parents, when they were in grad school, they were like, that revolution was like wrapping up, and they were like really working on the Cerrado. And when I talked to my mom, she was like, it's amazing, how much we can produce now in Brazil, in Cerrado, with all this technology that came through. So in this scenario, Embrapa was founded around a little bit more than 50 years ago with the mission that to generate knowledge and technologies for Brazilian agriculture. And we are having been working more and more on this. So, this is how Embrapa research stations looked like 50 years ago. So you can see that it's quite interesting, and was super important to have this knowledge and the scientists contributing. So Embrapa was sending scientists abroad, especially to US, to get their PhD, to learn more, and they would return to Brazil and applying this knowledge and transfer this to students. So that was a groundwork together with several different universities like Exalc. So this looks like a field plot for experimental station. Still, we can see a lot that looks like this. And this is how our agriculture looks nowadays. So it's big fields, lots of production, lots of technologies, cloning, seed technology. So we have advanced quite a lot. So this is more like a dream, let's say, field station looks like. So it's Maricopa in Arizona. So they have a field station with a different type of camera, scanner. So you can see that is in the middle of the desert, but you can phenotype with a lot of precision and different traits. So it's quite interesting. So that's the phenotype is quite important for any type of reading, right? So if you can check, you need to get from like far breathing, like from seeing like from above or inside the plant. So that's something that we need, we're gonna talk more about today. So if you compare, let's say, conventional breeding with this new technology, we are just basically trying to accelerate and get new traits into the breeding. So they're together. So nowadays, I don't think it makes sense to say that it's like one or another. They are just kind of, but together, in order for us to get into better and more productive crops. So basically, we can have to achieve this faster, incorporate new traits that were not there, but we have always to worry about this last part, because for breeding, people were like mixing, crops, but nobody really, let's say, cares about. But when, especially, I think you are all aware about the transgenic when it started, it was very polemic. So we have we need to fit a robust regulatory framework, and this costs a lot of money, and then you need more time. So you need to be aware that use of technology also has this other side that we need to care about. So why do we need new technologies? So why we are going to integrate omics and have different tools? Because we have this scenario where we have extreme events. We can see like heat, drought, waterlogging, and the frequency of these events are increasing in general. And they are also occurring simultaneously. So that's something that we need to work if we want to keep producing more. So that's an example in maize. So we have, with the heat events that we had in the past years, that we have a yield loss in yield production. So we need to worry about this because this is happening right now. It's not a solution that is fast. You can ask like any company what they are looking about. So it's to produce more. They are worried about drought. They're worried about new pests. So that's something that you solve one problem, it's gonna arrive another one. So we need to keep working. So that's where system biology comes to. So in this framework, we can actually have experimental design. It's very important, have a biological question. So what do we want to solve? What is our question that we're targeting? Have the hypothesis, and then we are going to use all the omics that are available and that our money can, can buy. So then we need to integrate a network. So modeling, computer power is super important for this type of technology. And then you need to link all this information that you are generating with the real actual phenotype. Because if you know like all the regulatory networks, but you don't know how is your phenotype there, I mean, you're not doing much, right? So that's very important to bridge this information. So when we talk about this omics integration, we are talking about getting into the phenomics, genomics, transcriptomics, proteomics, metabolomics, lipidomics, epigenomics, interotomics, and ionomics, bridge all this together, and then have a more tolerant to biotic and abiotic stress. So before actually entering in the work that we perform at Embrapa, I'm gonna touch down very briefly because I'm sure that during this week you're gonna see great talks and great examples of work that actually are applying these different knowledges and getting really nice results. So first one is phenomics. Second part is genomics. Then we are gonna go to the omics integration, transcriptomics, ionomics, and all. Prediction of phenotypes, and then finally get to a superior line. So just in short, these multi-omics technologies in synergy to revolutionize crop breeding through more precise, data-driven, and sustainable innovation. So that's what we want to get. I want for you to pay attention to these, especially students, because the experimental design, it's very important. So you can spend lots and lots of money to do thousands and thousands of omics, but if you don't have a great experimental design, then you're not you're not answering your question. So for this, you need to think about, which genotype you're going to use, which tissue, developmental stage, are you, evaluating any stress? How is your stress? Is your stress really a stress or not? Because if you're give a very severe, your plant's gonna die at the end. So you harvest, but then you don't have a plant. So that's a problem, right? Then you need to have the samplings, like protocols, what you're gonna use. Think about processing. So today it's a issue, right? We produce many data, and how you're gonna handle this data. Do you have a server? Did you talk with a bioinformatician to statistician? How is your group actually working? And then you have to pair these omics, because every time that we generate this, it's a lot of information, right? So transcriptomics, metabolites, genes, and then you're gonna reconstruct and join everything together. So I would talk a little bit about phenomics, because I think maybe that's one of the most important part, because for all the omics, you're gonna need the phenotype at some point. So phenotype could be very, simple, like checking them and say, oh, this is bigger, or this is blue, this is red. So it started very simple with traditional measurements, low data volume, and manual record. Then we went through 2.0 with simple sensors, low throughput, and manual data processing. Then we started to have platforms, high throughput, and offline data processing. And then we went to this multi-scale, ultra high throughput, and real-time measurements. But we also have some issues that we have to think about when we work with phenome. So basically, it's because realistic environment conditions, they are challenging. Sometimes you want to measure for drought stress, and then you have a disease. You have like maize stunt that will come and then kill your maize. Then my drought didn't really work, right? So that's an issue. Sometimes you have such a controlled conditions that it's difficult actually to transfer this to the real world. So that's something that you have to think about. Cost, work, and time efficiency. Sometimes, of course, you want to have, that platform very automatic, great. But then you don't have the money to pay, or it's difficult to maintain. But then you have students that they can do the measurement much cheaper, right? So, sorry, but that's true. So what happens? Then that's your reality. So you're going to do that and then have the same measurements, right? So that's something. And then you have the sufficient resolution. So if you're checking, for example, for a trait, do you need that information or that's too much? I'm going to show an example of a trait in a work that we perform different experiments, more complex, but the simplest one gave the same result. So why would you spend more money and be more complicated if you can get the same trait very easily, right? So that's something that we have to keep in mind. So when we talk about phenotype, we can go to plant scale, field scale, form scale, or country scale. So depends again on your question. When we talk about SARC, for example, that's a work that Embrapa does to monitor and actually drives where it's safe to plant, when it's safe to plant. You can go much more above. But if we're working for a trait that needs physiology, needs to know which nutrient, then you have to go into the plant scale. So this is just to exemplify that you have nowadays lots of options to phenotype, so depends on your question. So you have low cost, easy to measure. You have UAVs that nowadays are very easy to have and to manage. And then you have all the other platforms and tractors, so depends on your power of analysis. So one thing that we need to keep in mind here is that our ability to control the environment goes in the other hand than your phenotype depth. So you kind of need to choose what fits better in your model. So, okay, now I have our phenotype. We know how the plants look like. So now what we need to get? Well, we're going to need to get into the letters, right, to the genomics. So how would we access the genes? So from this, we can have three pillars for the genomics, basically functional genomics, structural genomics, and comparative genomics. All of them you can, at the end, get into the improved yield and growth. So for functional genomics, we're going to sequence, identify genes, then you can gene edit or overexpress, do some type of transformation, and then you have this transgenic into evaluation. So you can confirm your gene through this technique. Another one is to analyze and have KTL mapping or association mapping, where you can get into your trait, develop a molecular marker, and then use, and then also confirm. And the other part, you can identify orthologous genes, identify the network and the regulatory part, and then integrate the omics and also get into a gene. So they're all options. So when we talk about cultivated plant genomes, we started with Arabidopsis. That's not really a cultivated plant, but it was very important model plant. And then nowadays we have many more than these ones, but there are some examples. But it's not, we understood that it's not only important to have a reference genome, but also have the pangenomes. So pangenomes is starting after like 2000 have Arabidopsis, and now we have many more, because that's an example in soybean, because the pangenome will get different individuals so you can know what is the core genome, what are the genes that are common, and what are the accessory genomes. So the different genes that are there that maybe not, it's not that they are not important, but they are different among the different individuals, okay? So what for? Why would we do this? Because with this information, we can get into some genes, some domesticated genes, for example, that you can modify and actually accelerate and have a certain trait like higher grain yield, for example, or a different flavor, or a different starch proportion like amylose, for example, or you can use synthetic biology. So we have an interesting project that's the rice C4. I don't know if you heard of it, but it's to transform a rice that is a C3 plant into a C4. So they are knowing the genes and doing a lot of synthetic biology and construction in order to try this. Is going to work? I don't know, but they are making a very interesting progress on that hand. So with all that, we have different tools that we can apply to get better or crops or transcriptomics. So, okay, we have genes, but how about the gene expression? So we have different generations of that. So we started, years ago with the STs, microarrays. We have been done a lot of these. Philippe is laughing because he did a lot of that. Then we have first and second generation. So we are capable a lot of identifying genes and networks of expression. So nowadays, we are going more into the single-cell expression. So it's not only understand the general expression, but how each cell talks to each other, or how is this network. So basically what we can have is to understand each expression for each part, and then try to connect the expression with that. So that's a two works example where they use the single cell and identify genes that control development. So if you know that genes that control that development, you can modify or select and then have, for example, a year with more rows, for example, that will yield more. The other challenge is the epigenomics, right? So epigenomics it comes from the epi above in Greek. So it's not the DNA. So it's not a modification in DNA. So this was complicated enough, right? But then we got something that is not controlled by a mutation, but it's something about the chromatin remodeling. So how the environment and the conditions will modify the chromatin in certain time, and this will pass this information ahead. So that's a different challenge that we need to understand now, and we have tools. So here we have 3 examples. I like a lot the palm tree example when I saw Rob Bartisans talk about it, because they had this mantled oil tree phenotype that takes a lot of years for the tree to grow, and they just found out at the end. It was not a DNA mutation, so it was a genetic modification in the karma genes that gave this phenotype. So, see, that's a real issue that we have. It's not DNA. So DNA was like, all fine, but then we had this modification. So that's something that we need to care also about, okay? Proteomics, so here we go to the biology dogma, right? DNA, RNA, and now protein. So protein also, you have different methods to access, and it's cool because then you can see, where the proteins, where they're located, how to control this information. The same with metabolomics. Now we can get into like single cell for metabolomics or like general tissue. We can isolate with different techniques, and also we have several different equipments that cost a lot of money, but now we have services that can do, or you can do collaboration, but it's also very important to know, which metabolites are there. Lipidomics. So now we are looking into the lipids. So things are getting more and more complex. So I thought that was interesting because this is the number of citations for when you had genomics, proteomics, glycomics, and then lipidomics after 2010, you're having more and more information about. So if you're working, for example, with soybean peanuts, might be a very important trait for you to work on. So here's just an example of the pipeline for lipidomics. And here an example where you have different agricultural products that they had the lipid extracted, and then they characterized these lipids. So you can increase a certain lipid that is important. Anyway, lipids are very, very, very important for, for general brain, for example, formation. So that's something that's important to know. Ionomics. So Philippe is going to talk a lot about this. So well, basically, you can get into, ions are important, are fundamental. So you can get into the regulation, find genes, and then have, for example, one of the example that he works that he do that is like increase iron, for example, in rice, and you have many, many more examples on that. So with that, you need to join everything together, right? Because if you do all the omics, then you need to join the information. So here's a little bit of this idea that where you have the, let me see if I have this. Okay, transcriptomics, proteomics, metabolomics, then you associate everything, integrating like with artificial intelligence, genotype, phenotype information, then you get to trait improvement, food security, future crops, stress tolerance, and higher yields. So it's very important to associate both information. So now you have all the omics, and then you have your phenotype, and you have your genome. So genome-wide association, it's something powerful that you can use, right? So you're going to need to have your population selection, identify phenotype, genotype, associate everything, calculate, and then you're going to have a marker. It seems that it's simple to have a molecular marker, but a true molecular marker is not that easy. You need maybe unless you're very lucky, right? But otherwise, you're really going to need to prove, and then after you have this marker, you have to go back and then phenotype, use, and really see, measure the effect of that molecular marker. So that's something that's very important and very useful. So that's a general overview of this part. So basically, you're going to need your plant, your population, mapping population that can be different ones. So you have an example, like a real, NAM, magic, different populations. Then you're going to scan this, decide your genetic architecture. I can see Professor Augusto here, and other professors that work with this part that is very important. Then if you have a candidate gene, then you're going to go for validation, and then you have to go back. So it's you just kind of keep going, but it's a very interesting way where you can integrate everything. So I hope at the end of the week, I'll not say at the end of my talk because I'm just like giving this first overview, is that you're going to have many examples, and you are aware that it's possible to use omics to get to better crops. I'm going to show 2 quick examples, and then I'm going to talk a little bit about the work. Fine. So basically, this is a cotton project where they integrated phenomics, metabolomics, transcriptomics, epigenomics, and genomics in order to have a desired phenotype. The other example, this is an example from my colleague Andrea Evelyn from the Effort Center, but they are working with maize and sorghum in order to get to more resistant to drought crops. So they're using a lot of omics. They use that phenotype platform that I showed to you. They also doing a lot of transcriptomics and all to get into interesting genes. So I wanted to bring this because this costs money. So, and this depends on the size of the project that we're talking about. So I put like 3 general lines. Of course, this depends a lot from pilot to medium to large scale. So from 1 to 5 and up years. And this costs around if it is cheap, $2,000 to plus $8 million, right? So depends on the, how many omics do you want to, to get? Are you doing all the omics? How many traits are you working on? Personnel, field trials, and then we have to be aware that we need collaboration. You cannot do a huge project if you're not in a network. It's very pretentious, I think. You say, oh, I'm going to do all by myself. No, you need colleagues with different expertises. So that's home take-home. I'm not in the middle of the talk, but that's a take-home message that I want. Network. So here you have like different expertises that they need to go together, in order to get to a better crop. If you think about a company working with, maybe a few traits, but have different experts from different fields. So that's very important. And we have to be very conscious about, funding, right? Because sometimes you can have a great idea, but if you don't have funding for that, you're not going to do it. Then you need to work on something else.
How do you measure the output of those three sizes?
ChatGPT. Yeah, because I and I thought that was true because I know more or less.
I understand sizes, the costs, but I want you to understand what's the estimated output of each one of those.
What do you have out?
60 gene traits, 600 gene traits. What you add back from all this?
Sometimes it depends. Sometimes you can, in the first one, if you can be like very nailed, you're gonna have like a gene that will be in a cultivar that you're gonna license or a company is gonna use and then can reverse in a lot of money. I'm thinking about, like, for example, BT and Brapa that made the first national transgenic. It costed like 10 million, right? And you have one trait. So, it's worth it. I think it is because the companies are investing on that, but it costs money. And I think it's even back to society because if you didn't have, all this technology going through, our yield was not going to be the same. So just for this part, I'm going to talk a little bit how we manage all this work at Embrapa. So mainly my work is focused on phosphorus efficiency. So, okay, that's all right. So why we work on phosphorus? So if you can see these red parts is where you have low phosphorus availability. And Brazil is very dependent on phosphorus fertilizer import. So most of our phosphorus comes from abroad, especially Morocco. And the price, especially after the Russian-Ukrainian war, ramped up a lot. So we are always dependent on this market. So it's important for us to invest in technologies to get this phosphorus. Because phosphorus, actually, it's there, but it's not accessible to the plant. So phosphorus, it moves very slowly. It's different from nitrogen, for example. So you need the plant to get the phosphorus or, to interact with a microorganism, but somebody needs to get the phosphorus there. So it's a big challenge. So that's why we work with roots, right? Because we're talking about soil. So we need roots for water and for nutrients. That's the basic for a plant growth. So why did we decide to focus on roots? So phosphorus use efficiency has mainly two components. The P acquisition, that's how it gets from the soil, and the internal utilization, that is how phosphorus is used inside the plant. So our breeders, both for maize and for sorghum and maize, found out that we have 7% relative cost and 30% for internal utilization. So we decided to focus on the 70%. Not that the 30% is not important, it is, but that was one decision that we made. So we have 3 pillars to acquire phosphorus. And today I'm going to focus on root morphology and architecture, but we've also been working a lot with microorganisms and the plant-soil microorganism interaction. So for the root traits, I just want to highlight this work that was done together with my colleagues Cláudia and Jurandir Cícero, sorghum breeder from Embrapa, and different students. So I'm going to talk about this case that started with a KTL that's called phosphorus uptake 1 that was found out in rice. So what they found is that in a diversity population, they had a variety called Casala that had a much higher P uptake than IMPB, that is our reference genome. So this was much more efficient. And they had a rice neosogenic line that was carrying, when we was carrying the PUP1, it showed a threefold increase for P uptake and also an enhanced root surface area and yield. So on 2012, they found out that the gene responsible for this KTL was PISTOL1, or phosphorus starvation tolerance 1. So when they transformed the overexpressed this gene, they found out an enhanced root surface, enhanced P acquisition, and also yield. They also did this with a new population, so it's a very nice work. So we had a joint project together at that time. So what we did is that we are looking for the orthologous genes in sorghum using association and KTL mapping in a real population in maize. So for this, we needed to work on phenotyping, right? Because if you're talking about root traits, we need to develop phenotype methods. So we have been using a paper roll method where we basically put the plants in these folders. Then we photograph the system and analyze with WinRhizome. This was done also in Cornell, where we have a 3D phenotyping platform there. So we had these different containers with nutrient solution, and then we could photograph at 360 degrees. And then we have to go to the field, right? So that's the shovel omics. So I think the omics need to integrate that part. So basically you need to get the shovel and get lots of information about. You can see that can be quite challenging for us here in Brazil because we have a very clayey soil. But basically, we need to get that roots out. We photograph the root system, and then we need a system to evaluate. We have been using dirt. There's other options, but that's one my colleague Thiago from Embrapa Digital Agriculture helped us to put together. So that's showing the video. You know how much work. That's why. That's quite interesting. We have a truck that I saw in one of the root phenotyping meetings, that they have a tractor that comes with a tube and takes the plant out, but here don't have that, so we use students. So all my students, they're kind of, if they pass through a field with root phenotype, then they're okay. That's why they want that because, it's a lot of work. Get this out and then wash everything, photograph, get data, and all. And of course, you need field. So you need, nice experimental field, very well characterized. Because here you can see, sometimes you have, a phosphorus stain in the field, and this can influence your result if you're working with nitrogen. If you're working with, drought, but then you have, water leaking, then it's an issue. It seems that is, looks ridiculous, but it happens, right? So it's something that we need to care about. So what we did is that we got all these root traits, and then we thought it's a low but significant correlation with yield data. And with that, we went for association mapping. So we had phenotyping, yield, root data, phosphorus data, and then we had gene information. So what we showed that for one of these genes, when we had decrease in root diameter, we had an increase in P uptake. When we have an increase in root surface area, we have an increase in yield. So that's cool because that shows that allele substitution here leads to an increase of between 150 to 200 kilos per hectare. So you can see a real effect of a SNP, like a gene, like a nucleotide, to our actual result under field conditions. So this was performed here in Brazil and also in Africa for biomass. So we also used a panel that was phenotyped there. Important, we used 3 different populations. So we had S2, association mapping, and also a real population, and we found the same result. So it's interesting. Now these markers are already in our breeding program, running and giving positive results for selection. That's something that I really enjoyed. We published this last year. So here we have like 3D, 2D, performance, and field. And I love this because I have 2D data here that actually correlates with grain yield. So we did like different methods, but the simplest one that I was doing, and I'm proud of this because when I started at Embrapa, I started this method, and when I was talking to the breeders, they were not really believing me. And so when I see this, I was like, oh, you can actually measure and have a field result. And what is cool here that is found out, because we always have this thing about, maybe you heard about this, right? That phosphorus ideal phenotype is that umbrella root phenotyping, so more open. And for drought, you have narrow angle, deeper root. So what we found out, there are certain sorghum roots that they are more efficient because they are deeper, but you have more roots all over the profile. So what we did is that we had, to prove this, we had these big tubes, and then we have the soil from Cerrado region. So actually we get the real soil profile here, and then we measure the different amounts of roots. And we actually really saw that the most yield and efficient genotype actually had more roots here, but has more roots all over the soil profile. So that's quite cool. And then we kind of found ideal type for phosphorus and drought. So this work was done together with Professor Augusto is here in collaboration with him and the students at that time. So we had a real population for maize where we had a cross of efficient and inefficient genotype for phosphorus acquisition. So what we found here is that we have a KTL that colocalized with the efficient genotype, root, surface area, yield, and it was more expressed in the genotype where the allele was coming from. So with that, we had a couple of candidate genes. So what we did with that, we chose the rice candidate, the rice as a positive control. Then we had 3 maize candidates and 3 sorghum candidates. So we had a cassette. We transformed maize, did all the transformation steps, and then we came back to the phenotyping. So what we observed is that most of the genes had enhanced shoot dry weight and also surface area when they were overexpression, both under low and high peak conditions. When we went to the greenhouse, we also observed this effect, so with low and high phosphorus conditions. So you can see an increase in shoot dry weight and also root dry weight. We also evaluated this under field conditions. We are evaluating the crosses because it's very difficult to evaluate inbred lines under field conditions. But even here, we can see, here's our wild type control, and then we have our transgenic lines that they have higher, bigger root system in general. And we saw a positive result, so it's quite exciting that these genes are really having an effect under field conditions. So you see, we came through phenotyping, genotyping, mapping, then we got into the genes, then we transform lines, then we have to go back to the phenotyping to see the result, okay? We also did this with tobacco because we want to get a monocot and a dicot. Here we can see even a better effect actually in tobacco. So here's our negative controls, and here the different genes. You can see negative control, and here one of the genes. So bigger plant, bigger root surface area. Here we have the results under greenhouse conditions. So here we have also enhanced shoot dry weight under low and also under high peak conditions. So in this part, what we know is that maize and sorghum, Pistol-1 genes, they have similar roles than the rice Pistol-1 gene. That this gene has the potential to enhance root length, pea acquisition, and yield, and the markers can be used in breeding programs. So that's something cool, and that's something that I like about Embrapa, is that because we can do work and actually we can apply the markers into a real breeding program. So you can see this effect. So I think it's an advantage that we can have. So just to kind of wrap up and have some messages for you, omics, it unlocks the potential. So you have the molecular insights into plant responses. So a lot of some things that you see, but you cannot understand, when you do the omics, things get more clear. We need to bridge lab and field. So you need to translate this genomic discovery into the agronomic real world. So we need to do this both hands. Efficiency and resilience. So omics-based strategies enable crops to use resources more efficiently under climate stress. Sustainability at the core. So if you want to reduce, fertilizer use it more efficiently, because that's an issue that we have. Sometimes you apply a lot of fertilizer, but you don't have any yield increase. So you're spending money, you're having environment cost, and you're not getting anything out of it. So it's important to have this. Also water, it's a very precious source that we need to take care. Other one, collaboration is essential, right? Our progress depends on what we're doing here, right? Listening, learning, networking. So today we're gonna have at night. So it's important especially for us too, but for the students to get to know the researchers, to open their mind for different fields, what's next, what can do more. So that's very important. And future outlook. Omics will continue to guide the design of resilient, climate-smarted crops and sustainable farming system. So we need to, to work and learn more. There's a lot of data out of that we can use. So sometimes if we cannot really generate our own data, we can use the data that is available. So it's important to know that, right? So if we think and if you know the tools, we can do a lot on this end. So with that, I would like to thank all the group that I work, so our researchers. So I don't work alone at all. So a lot of this work, it's collaboration work that we have. The work today, especially, I have to thank a lot Claudia and Jurandir, that we work a lot together. That's my colleagues from microbiology, because I didn't talk about that today. Group of students from FCJ and also Unifem. My colleagues from GCRC, so Isabel, Dante, and Juliana that is here. Tiago, that helped a lot with the root phenotyping. The funding, of course, because with funding we don't do much. So I also need to thank them. And with that, I would like to thank you and take any questions.
Now we have time for questions.
So thank you very much, Sylvia, for your presentation and for sharing these very interesting results. My name is Alexandre Aono, and I've been working in Sweden since last year. And it's really difficult to work with roots, especially from the analysis part of it. And we tried to develop models to capture characteristics, very fancy models, deep learning stuff. But in the end, the most interesting and important trait that we obtained from those models was the length. Yeah. So, so when we were telling about it, it's like we had the same experience. So I would like to know, besides the length, besides like the amount of, let's say, pixels related to the root, what other characteristics do you think that have been underexplored? And what do you think would be like the direction for getting extra information on roots besides these traditional ones? Thank you.
You're welcome. So it's so challenging. I was in a root conference in Mizzou like a couple of months ago, and like everybody was talking about different phenotype methods. So there's a group that models, the one that have like these fancy platforms. I mean, so it depends, because for example, for Rhizotron, there is a nice network and they have like platforms in different places. But for maize roots, the correlation is like 0.3, so you cannot use it. But for rice or for barley, you can use, so that's fine. So depends a lot on the crops, but mainly I think is the root surface area in general and angle that might be the most important. Because sometimes we dissect too much, but it's so plastic, then you have like a little bit of drought. So the problem of controlled environment experiment with roots, because when the root touches the pot, it changes a lot already. So that's an issue that we had with that experiment, for example, because the roots, they go all down. When they start rolling. So then what do you consider? I think, so that's a discussion that we had. So it's, so I think, sometimes we have like the ideal, because when you have like NMR, for example, I was in Jülich in Germany, and they had that great platform there, but it's not high throughput, right? You can do just, a little bit at a time. So basically, I think you need volume. So, now, for example, for this population, it was like 400, different genotypes. Then you have to have all the replicates. So you're talking about thousands of roots. So you need basically something that you can do throughput. If you have money, you can do more like automatic. Or otherwise, you can do like hand by hand, and then check this trait. So we're being, we use different techniques. But at the end, if you check like all these populations, then the simplest one works at the best. So, yeah.
Nice. Thanks for the very nice talk. Very interesting work you're doing. So I'm interested in the ionomics aspect, of course, of what you're doing. So you've shown that you can increase phosphorus in those plants. And are you doing actual ionomics profiling in those samples and trying to see if there's any other micronutrient that has been affected?
No, we didn't. Actually, that's something that we kind of wanted to do at a certain point, because, but the thing that the cost was actually the bottleneck there, because first we were doing like phosphorus for everything, and then we couldn't do like all the nutrients. But I saw some work that they had a correlation with other nutrients with that, but we didn't have the information because we didn't do it, basically. But it would be great to do.
Congrats for your presentation. It's good to go to a congress like this to see other kind of opinion. What I have in my mind is that the cost of genotyping decreased along the time. But related to the slide that you show, related to the costs, it seems that's a little different, because depend of the size of the game you are want to play, right? In your view, we are going to have this kind of a figure for a long time, or are there any kind of things that we can do this for reduce this cost when you think in big use or integration of omics?
That's a good question. So, so depends on the problem is that I have the thought that for the simple problems we already kind of found a solution, but for complex problems, for let's say drought. Right? Everybody's working or worried about drought, but we don't have a magical solution. Because, you know, to be really resilient, oh, let's get a cactus, right? But then you don't have an yield. So how you're going to convert, you know, this maize into a cactus and yield? So, so that's, I think it's not only a trait that is going to solve a problem, but we need to look, you know, for the system. So that's the thing. And for the investment, I think we already, depends. We're going to need these big projects because the big network projects allow us to advance more. So that's something that I admire a lot. For example, in the US, they have like this big project that, you know, through different states where we have different expertise, but this costs money, right? And I think it's worth it to, you know, to invest in science because sometimes you don't have not necessarily one trait, but you're going to have different expertise and knowledge that are produced that will be applied in different places. So I'm not sure how funding for this, but I hope that we always going to have funding for science and tackle different problems. Because if you solve drought, then you have a different disease. Because if you plant, another disease is going to arrive. So that's, I think, luckily for us scientists, we're always going to have a problem to solve. Okay, thank you.